Eukaryotic DNA replication termination. This will be discussed in two parts. In this session, the termination that is going to be considered is uh, when the replication opens up at different origins in the linear DNA. How do the how does the termination take place once it originates from the origins of replication? And the second part will focus more on the fact that because DNA is a linear, DNA in eukaryotes is linear, the ends of the DNA have its primers removed but not filled up. And so there is what is called as an end replication problem. And that end replication problem is taken care of by a particular molecular mechanistic, which will be discussed in part 2. So let us look at the learning outcomes of this session. The replication eukaryotes is reported to take place in fork merging zones. So there are certain zones where the replication forks kind of merge together. So this is considered to be generally midway between two origins that have fired. Majority of terminations may or may not be sequence specific. The tilt is more towards being not sequence specific and are dependent on initiation stochasticity. What does that mean? The random probability of the origins firing in a DNA is what is understood by stochasticity, and depending on the firing of the origins, you would have termination accordingly. So at the sites where the replication forks converge, the replisome machinery is known to disassemble. When the forks begin to converge, there is a topological strain. However, it cannot be you cannot have supercoiling further on the non-replicated part of the DNA, as that has to be replicated, and therefore it is resolved. It is seen to be resolved by forming what is called as precatenants. And these precatenants are understood to be resolved by DNA topoisomerase, although its molecular mechanistics is not really cleared. One other important fact that is taken care of by the termination process is that there is no between two origins there is no DNA that is not synthesized. So the RNA primers, one removed, are sealed, are filled up and then sealed. And the replisome machinery disassembly or the removal of the replisome machinery has a pathway that is not observed in prokaryotes. So these are the things that we will understand as we progress with the session. Now, what does one really understand by fork convergence? So let's say, for example, you have your linear eukaryotic DNA, which has opened up at its origins randomly. And as one can notice, therefore, for every origin, you will have two replication forks, and both the replication forks will be moving in opposite directions, thereby making replication bidirectional. So let us consider what happens when the replication begins and continues to elongate. And after a point of time, as the two forks move in the two sides, so when you can see from this position, the fork has moved further, and this fork has also moved further. From this origin, this fork moves, and from this origin, this fork moves. So at a point, they would come almost encounter each other at a particular zone. So you will see that same thing happening over here. So these are considered to be what is called as the convergence of replication forks, and these are what are considered to be convergence zones. Now, there are several studies which indicate that the sequence that is present in the convergence zones have been found to be quite similar in several species, and therefore there are suggestions that there is a genetic aspect to that, which means that possibly there are certain sequences which would contribute to making that zone a convergence zone, but it is still not very clear. And what is important to note over here is that once you have the convergence zones and the replication forks converge, what is or how does termination occur over here? Because the idea is very clear that at the convergence zone you would have termination happening, and as you can see, it is generally midway between the two origins of replication. So we are now going to look at how termination at the convergence zone can happen. So let us consider that when you have the origins opening up, we all know that the entire replisome machinery is assembled at the origins thanks to the licensing and the firing. And then after post firing, you know that the CMG helicase becomes active and the DNA polymerases are attached to the CMG complex. Both DNA polymerases, that is DNA polymerase epsilon, which is synthesizing the leading strand, and DNA polymerase delta, which synthesizes the lagging strand. And the plus you have what is called as the DNA primase complex. That too is associated with the lagging strand. As in the lagging strand, primers have to be made for every Okazaki fragment. So that basically constitutes the replisome machinery. Just roughly showed that there is a machinery over here and there is a machinery over here, with the emphasis being that the CMG being, that is the CDC45, MCM27 and GINS being the complex to which most of the other molecules of the replisome are attached, whether it is RPA, whether it is RFC, anything that comes and binds to this CMG complex itself. So, from this point on, therefore, what we understand is that the replication fork is ready to move ahead along with the replisome machinery carrying out DNA replication. So you will find that one of the strands is considered to be the leading strand. It is synthesized continuously, while the other strand, you can see that primers are formed and they are synthesized in fragments. So this, for this replication fork, this is the leading strand and this is the lagging strand. While for this replication fork, this is the leading strand and this becomes the lagging strand. Now, as the two replisome machineries converge, they come more towards each other. This portion, which is the unreplicated DNA, has to be replicated, and here overwinding is not feasible any longer. So, to compensate for the strain that is created here because of the helicase opening up the DNA, one finds that one of the ways by which the eukaryotic DNA takes care of the strain, the topological strain that is created here, is by forming precatenants. So you would have catenation happening in the replicated DNA, so that this strain that is formed over here is taken care of by the topological changes at other portions. So you will find that the eukaryotic DNA, as it progresses with replication, there would be catenants present at several points, again stochastically. So this is something that happens in the eukaryotic DNA replication. There are several reports that have suggested formation of precatenants, and also catenants at the termination site itself. Now, we still have to look at how termination happens. So, what we will basically understand over here is that once you have the replisomes coming towards each other, what is it that is going to happen to the two so that replication is completed and also this disassembles? Because we know that replication termination is associated with the fact that the entire replisome machinery has to disassemble. So there are several theories that were proposed. One of the theories was that as these two further come close to each other and encounter each other, when the two CMG complexes encounter each other, this interaction of the two CMG complexes can change their conformation and lead to the disassembly of both simultaneously. That was one proposal. The second proposal that was there is that maybe one of the replisome unloads first and then the second replisome unloads. And the third proposal was that neither of the two unloads at this convergence zones, they just bypass each other. So several studies in yeast, frogs, etc. have led to a clarity on the fact that the third proposal is what is happening, and that is convergence bypass. So extremely important to ask over here, and what we discussed is what happens to the replisome machinery at the convergence zone. So, like I mentioned, several studies have shown that this machinery will continue to move on the continue to synthesize the leading strand using this one as the template. And we all know that the CMG complex is actually binding to a double strand but a single strand. That means it needs both a single strand and as well as the double strand. So this complex will continue to synthesize this template, while this complex will continue to synthesize this template. So effectively what you observe is that at this convergence zone, the two replisome machineries bypass each other, and therefore this is what is understood to be convergence bypass. So this has been attributed to the fact that unlike in prokaryotes, where you have the DNA polymerase 3, which is a dimer, and both the monomers or the core enzymes of the same enzyme are associated with the two template strands. And it is that one complex that is synthesizing both the leading strand and the lagging strand. However, in the eukaryotes, it's not the same. You have one DNA polymerase that is synthesizing the leading strand, and you have another polymerase, which is the DNA polymerase delta, that is synthesizing the lagging strand. Moreover, the DNA pol delta also is associated mainly with the CMG, and the CMG is reported. It is proved that the CMG is associated with the leading strand and not with the lagging strand. So, by all means, the CMG can continue to be attached to the template which is synthesizing the leading strand. So thereby, in eukaryotes, this possibility is more rather than in the prokaryotes, because of the way the replisome machinery is associating with the template strands. Now, therefore, what happens is that the lagging strand of the replication fork of this, okay, is now being used by this replisome machinery to continue to synthesize the DNA till the entire replisome machinery reaches the primer of the Okazaki fragment, the immediate adjacent Okazaki fragment. So you will find this doing it in this template and this replisome doing it on this template. So you can see that from the convergence zone itself, the two replisome machineries continue to synthesize the DNA as long as the single stranded template is available. So that is something that is happening. You can see that the DNA is being synthesized. Now, therefore, you must understand that it continues to synthesize until it reaches the next Okazaki fragment. Now, what happens next? So, once the CMG complex along with the DNA polymerases reaches the primer of the Okazaki fragment, this primer is displaced to form a flap, okay, and then the Fen1 is recruited by the CMG complex, or which is already bound to the CMG complex, is able to fragment the entire flap. So the RNA primer portion is therefore removed, and once that is removed, okay, the same DNA pol delta continues to synthesize, that is fill up the gap that is formed because of the RNA primer being removed. And then once it fills up the gap, so this gap is filled up by this replisome machinery, and this gap is filled up by this replisome machinery, okay. The only thing that has to be done is sealing the gaps, and the gaps are sealed by the DNA ligase, which is also associated or recruited by the CMG complex itself through RFC. Through PCNA, because PCNA is also bound to the CMG complex through RFC. Now, therefore, what is observed is that the leading and the lagging strands have become continuous due to the efforts of the replisome machinery. So now, once you look at this, previously what you have looked at is that the replisome machinery is partly bound to the single strand DNA that acts as the template and to the double stranded DNA that it is synthesizing. But now, once the entire leading and the lagging strand become continuous and you have a complete double strand DNA formed, the replisome is now bound to a double strand DNA. So once you have the entire replisome machinery on the two strands bound to the double-stranded DNA, you have the next key event in the termination of replication, and that is unloading of the replisome. So what is observed is that the CMG complex per se is ubiquitinated, and the ubiquitination begins with a ubiquitinated E3 DNA ligase complex coming and binding to the MCM27. So once MCM27 is ubiquitinated, okay, and it has been observed that in the yeast it is what is called as the SCF complex that binds to the CMG complex through the MCM27, while in case of the frogs and in some higher eukaryotes also, there is the CRL complex with C standing for what is called as the Cullen domain. And so this domain is responsible for recognizing a lysine residue in the MCM27 complex and ubiquitinating it. So this event is extremely important, and now what you have is that the CMG complex is ubiquitinated. Now this ubiquitinated CMG is able to, only when it is ubiquitinated, it is able to recruit a segregase, and that segregase is having an inherent ATPase activity. And this molecule is also considered called to be as P97 as studied in several systems. In yeast, especially, it is called as P97. And this segregase or this molecule called P97 is able to utilize the ATP, and the energy released is used to unload the CMG complex from the double-stranded DNA. So it is basically this segregase molecules that unloads the replisome. Now, all the other molecules that are associated with the DNA helicase or the CMG helicase automatically, without any other molecular or without any other exogenous molecule present, just simply disassemble from the CMG helicase complex. And therefore, you will find that the entire replisome machinery is now dissociated. Here, what two things are important, and one has to note that one, as observed, an MCM7 is reported to be ubiquitinated. So this is 2 to 7 is reported to be ubiquitinated once the CMG is bound to a double-stranded DNA after the filling and the sealing is done. And this is important. Why is this important? Because if the CMG is ubiquitinated before the filling and the sealing is done, that means before the leading and the lagging strand become continuous, then it could lead to fork stalling and breakage. So it has to complete the synthesis on both the template strands, and only then CMG is ubiquitinated. So premature CMG is not good, and hence this fact that the CMG can be ubiquitinated only when it is bound to double-stranded DNA takes care of premature CMG removal. Now, after all of this, what remains is the catenated regions in the DNA, and these catenated regions in the eukaryotic DNA are decatenated to form finally the two nascent double-stranded DNA. Now, in prokaryotes, there are a lot of reports that have proved that it is DNA topoisomerases, and especially DNA topoisomerase 2, which carries out the decatenation process. In several yeasts too, it has been observed that it is DNA topoisomerase 2 that does the job. However, a definitive molecular mechanistic is still not understood in the eukaryotic system, especially in the higher eukaryotic systems, except for the fact that decatenation does happen to give rise to two nascent DNA molecules. Now, there are several reports that also suggest that termination can be site specific, and in certain genes, in certain regions of the DNA, eukaryotic DNA, it has been found that termination is site specific. So one of the examples that is there is that of the rDNA locus. So it is that part of the DNA which gives rise to rRNA. Okay, and there what has been observed that termination happens by what is called as sequence-specific replication fork barriers. Now, what is this? This is very similar to the fork barrier in prokaryotes that is formed by the ter-tus complex. So, what is observed in these regions where termination happens is presence of certain cluster of tandem repeats. So, that forms what is called as the cis element, and that is named as a ter element, and these star elements are bound to by a terminator protein, FOB1. This has been studied especially in the budding yeasts, and this FOB1 can bind to the ter element to form what is called as a polar replication fork barrier. So therefore, effectively what is understood is, suppose this is one replication fork coming in and this is the other replication fork coming in, and this is what is the convergence zone where the termination is to happen. Then in such termination process where there is site specificity, what is observed is that for a specific replication fork, you may have a ter region present over here. This ter element is bound to by FOB1. So you can see again over here, because of the orientation in which the FOB1 is binding, for this replication fork, this is a stop, this is a barrier. So you cannot have this replication fork moving forward anymore, and so you consider the fork has been arrested at this point. But for this replication fork, this FOB1 is not a barrier and it can continue to move. Okay, so by far what is understood is that this replication fork stalls because of the presence of the ter FOB1 interaction, while this replication fork continues to move, and as it continues to move, it will converge with this fork. Now, when there is convergence over here, again the studies have suggested that there can be convergence bypass. So you can have this replisome machinery present over here continue to synthesize this template, and the replisome complex present over here can continue to synthesize this portion from here on. So therefore, there is a convergence bypass that happens over here and replication concludes. So the conclusions that we make from this session therefore is that the replication forks, when encounter at the convergence zone, the replisome machinery being attached to the leading strand are able to bypass each other and continue to move. The replisome will continue to synthesize DNA on the strand until it reaches an Okazaki fragment, post which the replisome recruits FEN1 and ligase to remove the primer and fill up the gap and seal it. The CMG complex is now bound to a double-stranded DNA, and then when it is bound to double-stranded DNA, it is ubiquitinated, unloaded, and then the DNA decatenated, thereby taking care of termination. Termination in certain cases can be site-specific, like the tortoise complex of the prokaryotes, where a polar replication fork barrier is formed to stall a replication fork and wait for the other to converge. Thus, termination of eukaryotic replication happens almost midway between two origins when the replication forks converge, complete synthesis, and then unload the replisomes, ensuring that nothing leads to genomic instability. Thank you.